In the last video, we learned why spatial transcriptomics matters. Today, we'll dive into MERFISH, the method we use to map RNAs in the honeybee brain with single molecule precision. By the end of this video, you should be able to 1. Explain how MERFISH identifies many RNA species in situ. 2. Describe the key steps in the MERFISH experimental workflow. 3. Understand how barcoding and sequential imaging work together for error-robust detection. MERFISH is an imaging-based spatial transcriptomics method. Instead of extracting RNA for sequencing, we keep it inside the tissue, label it with special probes, and then use multiple rounds of imaging to read out unique barcodes for each gene. Each barcode is designed so that even if some spots are missing or an imaging round has errors, we can still identify the correct gene. The key to MERFISH is the encoding probe. Each probe has 2 parts, a targeting sequence that binds to the mRNA of interest, and a readout sequence that doesn't bind to RNA directly but will be detected later through a separate readout probe. We design multiple probes per gene to boost signal and make detection more robust. We assign each gene a unique binary barcode. A 1 means a fluorescent spot should appear in a given imaging round, while a 0 means no spot. By spacing barcodes far apart in Hamming distance, MERFISH can correct errors, so even if one round fails, the gene can still be identified. Here's how the experiment works from start to finish. 1. Fixation, preserving tissue structure and RNA integrity using 4% paraformaldehyde. 2. Permeabilization using ethanol and detergents to make the tissue accessible to probes. 3. Encoding probe hybridization, binding the encoding probes to their RNA targets. 4. Expandable gel embedding, anchoring the RNAs to a swellable gel mesh. 5. Digestion, using proteinase K to clear proteins and improve expansion. 6. Expansion microscopy, physically swelling the gel, increasing separation between molecules for clearer imaging. 7th, re-embedding in non-expandable gel, stabilizing the sample for many imaging rounds. 8. Nuclear staining, visualizing nuclei to help with cell segmentation. 9. Readout cycles. For each round, hybridize a readout probe, image the sample, strip the probe, and repeat until all bits in the barcode are read. In the microscope, each RNA appears as a diffraction-limited fluorescent spot. Across multiple rounds, the spots on-off pattern matches a specific barcode in the codebook. Once decoding is complete, we have the gene identity and precise location for every RNA molecule in the tissue. MERFISH gives us subcellular resolution, so we can see RNAs in dendrites, axons, and cell bodies. In the honeybee brain, this allows us to map complex structures like mushroom bodies, optic lobes, and antennal lobes, and to study how gene expression relates to behavior. In the next video, we'll walk through the workflow in the lab. You'll see each step from tissue fixation all the way to imaging with explanations of why we do it that way. This will prepare you for your own hands-on rotation work.